What is RNAi? Eukaryotic cells utilize multiple complexes and tightly regulated pathways to control gene expression. Among these, RNA interference, RNAi, is a natural process triggered by double-stranded RNA that leads to the sequence-specific degradation of mRNA, effectively silencing gene expression in many organisms, including plants, animals, and fungi. In this video, I will introduce the core concepts of RNAi in plant research, with a focus on a key class of small RNA molecules, small interfering RNAs. Small interfering RNAs originate from either exogenous sources or endogenous sources. In plants, they primarily function in post-transcriptional gene silencing and play a crucial role in defense against viral infections and modulating gene activity. Through RNAi or small interfering RNAs mediated knockdown, researchers can reduce the expression of specific proteins to study their function, for instance, by observing how cells respond in terms of morphology, survival, or behavior when a particular protein is silenced. Beyond its biological role, RNAi has become an essential tool in laboratory research and holds promise as a future therapeutic strategy. History of RNAi. Let's take a quick journey through the discovery and development of RNA interference. RNAi may seem like a modern biotechnology, but it was first observed as a strange phenomenon in petunias in the early 1990s. Researchers were trying to deepen flower color by introducing extra pigment genes, but surprisingly, the flowers lost color instead. This unexpected gene silencing was called co-suppression. Then, in 1998, Andrew Fire and Craig Mello made a groundbreaking discovery in the roundworm C. elegans. They showed that double-stranded RNA could specifically silence genes. Soon after, in the early 2000s, scientists realized that RNAi was not unique to worms. It was found to be a conserved mechanism in plants, animals, and even fungi. Finally, in 2006, Fire and Mello received the Nobel Prize in Physiology or Medicine for their discovery. RNAi applications in agriculture, medicine, and beyond. How RNA interference is being used beyond basic research. RNA interference isn't just a lab tool, it's become a powerful applied technology in agriculture, medicine, and research. In agriculture, RNAi is used to develop virus-resistant crops, like papaya resistant to ring spot virus. Provide precise pest control through a method called host-induced gene silencing, where plants produce double-stranded RNA that targets essential genes in insect pests like corn rootworm. In medicine, RNAi is being explored as a therapy, with siRNA-based drugs developed or approved for genetic disorders, viral infections, and cancers. In research, RNAi is a key tool for gene knockdown, enabling scientists to study gene function by silencing specific genes. Laboratory construction of small interfering RNA constructs. Let's look at how we design RNAi constructs in the lab to trigger gene silencing in plants. The most common and effective strategy is to create a hairpin RNA molecule. To do this, we clone the target gene fragment twice, once in the sense orientation and again in the antisense orientation, with a spacer or intron in between. A frequently used spacer is the PDK intron, which improves processing and enhances silencing efficiency. To drive high expression of the hairpin RNA, we use a strong constitutive promoter, typically the cauliflower mosaic virus 35S promoter. To successfully trigger RNAi in plants, scientists use specially designed plasmid vectors for carrying the genetic information needed to produce hairpin RNAs inside the plant. Some of the most commonly used RNAi vectors include P. Hellsgate series, PFGC 5941, P. cannibal. These vectors are typically introduced into agrobacterium, which then delivers them into the plant genome. A key method in many of these vectors is gateway cloning, a recombination-based system that allows quick and efficient insertion of gene fragments into vectors without relying on restriction enzymes and ligation. Why is gateway cloning preferred for RNAi vector construction? Gateway cloning is faster, efficient and high throughput, more flexible, and ideal for producing complex constructs like hairpin RNAs, making it the go-to method for RNAi vector construction in plants. Now let's walk through how small interfering RNA functions at the molecular level to silence genes. The stably delivering of RNAi construct into plant genome is typically done using agrobacterium. The process begins when part of its tDNA containing RNAi construct is inserted inside the plant genome. These plants then continuously produce double-stranded RNA and small interfering RNAs internally in their nucleus. When transcribed, this structure folds back on itself, forming a double-stranded RNA hairpin, which then exits the nucleus and enters the cytoplasm. In the cytoplasm, this double-stranded RNA is recognized and cleaved by dicer, an enzyme from the RNase 3 family, into small fragments of about 21 nucleotides. These fragments are known as small interfering RNAs. The small interfering RNA's duplex is then loaded into a protein complex called the RNA-induced silencing complex, or RISC. This is a multi-protein complex critical for gene silencing. In the RISC, the siRNA duplex is unwound into guide strand and passenger strand by the enzyme called argonaute. Argonaute has endonuclease activity and cleaves the passenger strand. This cleavage promotes the release and degradation of the passenger strand. After this, only the guide strand remains bound to argonaute, forming the active RISC complex. The small interfering RNAs acts as a guide inside RISC, directing the complex to any mRNA that has a complementary sequence. Within the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The processed mRNA is then transported out of the nucleus into the cytoplasm, where small RNA molecules can exert their gene silencing effects. Once bound, argonaute slices the mRNA, preventing it from being translated into protein. This results in effective post-transcriptional gene silencing, which is sequence-specific, meaning only mRNAs with perfect or near-perfect complementarity are targeted. Regeneration of transgenic plants. Tissue explants grown on selection medium. Only cells with acquired DNA from the bacteria can proliferate. Tissue explants are transferred to media containing different hormones to promote shoot and root growth. Once shoot and root have grown, plantlets can be transferred to soil. The plant will then be analyzed to confirm gene incorporation and function. Techniques to verify gene knockdown in RNAi experiments. Once we introduce an RNAi construct into a plant, we need to confirm whether gene silencing has actually occurred. This is critical to validate the success of the experiment. There are several methods available, depending on the level of gene expression being measured. qRT-PCR is the most common technique to measure the reduction in target mRNA levels. If the RNAi construct is working, we expect to see a significant drop in transcript abundance. Northern blotting is more classical but still valuable, especially for detecting siRNAs themselves or verifying the size and presence of the target mRNA. If the goal is to reduce a protein product, then Western blotting is used to detect changes at the protein level. Phenotypic analysis is another way to confirm silencing. For example, if you silence a gene involved in pigmentation, you may visibly see altered leaf color. Finally, small RNA sequencing can be performed to detect and map the siRNAs produced, confirming that the RNAi machinery is generating the correct small RNAs. I hope you enjoyed this video.